Okay, let's take a look at questions. When interbreeding is possible, those organisms are considered as the same species. Yeah, that's the definition of a biological species concept, right? But as we have seen last week, lions and tigers can mate and reproduce and offer offsprings as a ligers or tiger. So the question was, does that mean then they are the same species? If not, why? So it has been, I'm not sure whether I briefly mentioned this thing or not before, but this was happened because before they have actually realized that tigers and lions can make and produce this viable offspring, but historically they've been living in a remote, separate, isolated habitats, and they would never naturally mate to each other. Whenever they meet each other, rather than trying the mating, they will fight. So those scientists at the time obviously thought that tigers and lions obviously are two different. Their appearances are two different too. So it has been traditionally already fixed as separate species. But actually, they should be, according to, if we are going to apply this, the definition of a biological species concept, of course, tigers and lions should be part of, or members of one same species. That's right. But it has been that way for a long time already, traditionally. That's why we cannot really change those things anymore. Another very similar thing is in the case of this, the Nean, Neander, I'm not sure the spelling, Neanderthals, yeah. These ancient humans. For long, these Neanderthals have been considered to be totally separate species from our own, the Homo sapien, even having two different scientific names, Homo sapiens, us, and Homo neanderthalensis. But it turns out that at the time, our ancestors, Homo sapiens ancestors and Neanderthals actually interbred. So in other words, we actually carry their legacy in our genes because of such the history of this genes exchange through mating. So Neanderthals and our Homo, our own species, the ancestors, were actually indeed engaged in their mating, interbreeding. That means we cannot really consider Neanderthal as a different species than us. We should consider as the same species as these Neanderthals, based upon this biological species concept. Yes, it is. But traditionally it has been already, and some move to change this thing, to correct this thing, has been there recently. So some people actually consider this Neanderthal as like in these days Homo sapiens neanderthalensis, as well, whereas we Homo sapiens sapiens. That is a like putting subspecies name. So it's kind of a shade of a gray area. And strictly speaking, obviously, Neanderthals should also be considered as the same species as us. If that, like, consider this interbreeding thing as one key criteria for separating species, then obviously we cannot separate us from these Neanderthals. Uh, like that, okay? Hydrogen bond, hydrophilic, hydrophobic, LDL, collection of cholesterol. But why is it hydrophilic? Yeah, good question. Yeah. Later, actually, we don't have any chance. But when you, if you have a chance to take the biology, real biology too, then obviously you will get to know that clearly. So here in this situation of the cholesterol is, like I said already, is a hydrophobic molecule because it's a lipid. Okay? And it's a technical problem of delivering this hydrophobic cholesterol from liver to other part of our body through bloodstream. That's a huge challenge technically. So what do we do? The solution came up with that us was cover this individual cholesterol molecule. Let's say each cholesterol, like 10,000 different cholesterol molecules. These are something like 10,000 cholesterol molecule. Then, well, how about we cover this cholesterol on the surface like a sugar coating, like this, as a protein. So on the surface, there's a protein. And this proteins in nature, by nature, as a hydrophilic protein. And this protein, obviously, as I mentioned, protein has always some area of hydrophobic and some other area of hydrophilic. So this underneath of this protein can be hydrophobic so that they can grab this hydrophobic cholesterol. So it can function as a coating, cholesterol coating. So what do we call this then? Now LDL altogether. LDL stands for low density lipoprotein. Here the important thing is lipoprotein. Lipoprotein is lipid plus protein is a lipoprotein. So here the protein part is in green over here and lipid part in red dots. So cholesterols got together and covered with some proteins are actually LDL. So in this way, it is really, really convenient for transporting through blood because surface hydrophilic protein can take care of all these nasty water things. Inside of this LDL, all these cholesterol molecule can be now safe until they reach to actual final destination at the doorstep of each different tissues. But when they arrive at the door of each tissues, then the opposite problem occurs. Guess what? This whole cell is hydrophobic, cell surface. Cell membrane is hydrophobic in nature. So hydrophilic molecule cannot penetrate easily. LDL is now, because of this forming the LDL structure thing, it helps transporting them through the watery blood, but now it has the opposite problem of entering. When they try to enter into inside of the cell, now they have a big problem. Because this hydrophobia is big to begin with. That's one no. And on top of that, it is hydrophilic on the surface. So there's no way this LDL can penetrate. And this inside component, the ingredient, which is cholesterols, are extremely essential for each cell. Cholesterol is not something that we should avoid or anything like that. Without cholesterol, we die. So we have to import cholesterol. So how do we do that? Through transport, we create some kind of special delivery form called LDL, right? But now, one quick, easy solution, intuitively, one might come up with is, hey, disassemble this over here. Disassemble an individual cholesterol. Now, everyone go in at your as your free will. Yeah, that will work. But for some reason, cell don't do that. So instead, what's alternative? If they cannot do that, or what other alternative can they think of? What else? Receptors. So specific receptors. So this is something that every animal cell has to have on their surface of the cell surface abundantly. This so-called LDL receptor specifically recognize this LDL and then import, grab, and hey, let's go together. So they need some specific receptor because of this. So that's what this LDL and LDL receptor. Every animal cell has to have LDL receptor because of this. Without it, LDL cannot go into because of this chemical nature. Why does heat change ice into water again? It's breaking. The ice in ice state, every water molecule is now having all maximum number of hydrogen bond. Every water molecule has the hydrogen bond. Unlike in the state of water, liquid water, some have this hydrogen bond momentarily, and some others are breaking them momentarily, like chaotic status, and that's why they are enjoying the water, the status of a liquid. But at the solid ice state, every water molecule has this. How can we break? Putting some energy. To to break the bond, you have to have some certain amount of energy, like heating. Why heating can break some chemical bond? High temperature. High temperature can increase the movement, the speed of a molecule. So this molecule cannot be held, put because of their moving speed is too fast right now. So that's why bond breaks. So that's what we casually call, okay, in order to break some bond, the chemical bond, we have to put some energy. Heat is such form of energy too. That's why this hydrogen bond is being broken. DNA is highly negative charge, right? Because of this phosphate group, phosphate, like ATP, like individual component generating this nucleic acid, all have this phosphate group, phosphorus and oxygen, and this oxygen has two negatively charged area. It's a highly charged molecule. That's why DNA and RNAs are highly charged and negatively. So in order to have some specific interaction, some specific proteins with the positively charged area can interact. And that's actually a very important function in the regulating and also stabilizing genes. But the question is protein being negatively charged every time? Some. So proteins are all different. Some proteins are specifically very, very high in their positive charge. Some proteins are very, very high in negative charge. So in this occasion, the protein should be positively charged so that they can have a positive and negative interactions. And different proteins have a different characteristics. So some proteins are very high in positive charge. Some other proteins are high in negative charge, depending on their different components. So I don't know whoever created this. I have nothing but admiration for this the author of such a true loyalty for this your alma mater school. Wow, that's good. So that you consider yourself as a covalently attached with your school alma mater. Great. Yeah, I wish I could also follow your such enthusiasm when I was a student too. Well, I envy you. Well, yeah, really, seriously. I'm not joking. Uh, I didn't know actually there was something like a hydrogen day. I don't know why this particular November 2nd is a hydrogen day. I don't know. Try to guess. Maybe some element making sense. But anyway, good. Good to know. Thanks. How can one inject glucose including C14, into human body? So, several different ways. You can even you can synthesize glucose specifically. Not every carbon of glucose, but only some carbon consists of C14. You can generate, you can make, artificially synthesize such radioactive glucose, and then try to feed this patient. Hey, why don't you have this candy, C14 candy? It will be great. But that's very primitive way of. These days, actually, a cleaner way of not even using very small, tiny amount of radioactivity, which is not even related with the C14, and have attached it to this glucose molecule. There is a specific way, and trying to inject them into the through needle, through your blood, and then it will travel and then end up in wherever they are needed. So several different ways you can do it. Can partial charge be generated even by unshared electron pairs of the element which has a higher electron negativity? One thing is this partial charge was not due to the activity of electrons, but this partial charges, the cause for this partial charges was actually nucleus, isn't it? Some nucleus of an element has a much stronger desire for attracting electron, whereas some other nucleus of an element has a weaker desire for attracting the electron. That's why this electron negativity differences is created. So try not to be confused with this electron sharing, but the partial charges can always be generated by unequal movement of electrons. If electrons are moving unequal, this is a very difficult concept. Actually, I should be ashamed of myself by trying to explain this thing as if I know this thing. No, I shouldn't know, but because this should only be explained by quantum mechanics. How am I supposed to understand quantum mechanics? No way. We all actually, without fully understanding quantum mechanics, we can only pretend that we are knowing something. We are trying to represent our interpreted version of these things. That's the limitation. But having said that, so within our limited knowledge, what we can explain this is something, this partial charge, the cause, the fundamental thing of this partial charge is on the surface, it is due to the unequal movement of, unequal distribution, should I say more correctly, unequal distribution of electrons. And the reason for such unequal distribution is not the electron themselves, but usually rather for unequal power of attracting these electrons from the nucleus of an element. What is the difference between polar and non-polar bond? Same thing. Polar and non-polar bond usually only apply to this covalent bond, in that because ionic bonds are already polar bond. Van der Waals bond is already polar kind of interaction, but covalent bonds are supposed to be non-polar, but in reality, navigation sounds. I know that. I know that navigation sound. Are you driving something here? This non-polar things happens because of this unequal distribution of electrons. So can I distinguish them by whether it has an oxygen, nitrogen, carbon or not? Yes, actually. In practically speaking, yes. oxygen and nitrogen will definitely always create such unequal distribution of shared electrons. Yeah. And because of that, somebody will be unhappy. So this polar things will definitely create other partial the byproducts such as hydrogen bond. So by doing so, you can predict, oh, these guys will have some rooms, the chances of such polarity. Right. Carbon, on the other hand, is not going to. Carbon will not create such unequal attractions, so you can be safely consider carbon is harmless. When a radioactive element decays, the mass decreases. Where did the mass loss? Usually the mass itself, some of the mass loss will actually go into some difference. Some will become electrons, very rare. Some will become protons. So that's why this course of decay, totally different identity of element will emerge. For example, carbon, C14 will become nitrogen instead of a carbon. Is the reason why, because of this, the energy conversion and also their weight dissipation. How can radioactivity carry out in cancer in some occasions? This radioactivity is high energy, strong energy. The reason why radioactivity occurs is emit the energy is because it's too much energy for them. And such too much energy will damage our very delicate structure, like DNA. And cancer is always something to do with the mutation. The reason why we develop cancer is certain genes are changed through mutation. So, as a consequence, the gene's product, gene's function is changed into something we call abnormal, which I usually try to avoid to use that vocabulary. But in the case of cancer, I will say abnormal. So, because of this, the high energy will break or damage some structure of DNA and causing the cancer, the mutation, and as a consequence, the function of this gene production will be altered, and that's how we develop cancer. Just curious, I don't know if this is a good question. Yeah, I think it's a very good question because I cannot even answer this one. Such a ghosts have no way of receiving energy. I'm not sure actually. How I don't know about ghosts. So, are you sure that ghosts have no way of receiving energy? What if ghosts can receive energy? That's something totally your premise is not correct. So let's say let's assume that ghosts have no way of receiving energy. So ghosts are scientifically difficult to exist. Oh, is anything that cannot be defined by energy transaction? And it's something that's scientifically very difficult to explain. That one is also, I don't know really, energy. All those, in the field of science, all those material can have some capacity of generating and receiving the energy. But just because one identity cannot display such ability, it cannot exist in science. That one also I don't know. So uh, makes me think for a long time, for the rest of my life probably. Your question will bother me for the rest of my life. Thanks to that, but I will keep thinking about that. Thank you. So. Don't be, please just do more about this kind of difficult questions. So just bother me. That's all you do, all you can do. And I really appreciate and also feel sorry for your pain of trying so hard in this shitty class. But that's all I can say. Liposome. This is something that I'm going to cover in the later section of this thing, when we get to the lipid part, I'm going to talk about this liposome. So let's just wait until this one is also. So whoever had this question, she knows something about this chemical. The carbon, I have mentioned that in the classroom, carbon only this double bond or single bond, right? But in fact, that carbon also can participate in triple. For example, in the case with nitrogen, carbon, there are three covalent bonds like this. So carbon so versatile in that not only one covalent but double bonds are very common, and in many occasions even triple bond. But then will somebody will ask, okay, then why not this? Is it possible or not? And she assumed that it is impossible. So the question was, why is it impossible? Because if carbon has a ability to have, carbon has a four valence electron so that they can share four, so that, how about this having these four connections? Why not this? Right. For a long time, actually, this is impossible. It has been known as impossible. Carbon doesn't do that. So this morning, and there is a reason actually specifically. Why? Because in doing this, let me say this. Actually, if this carbon quadruple bond ever exists, then it has to be in this way. So another bond is not even through direct covalent bond, but this indirect bond, and then this quadruple bonds is possible, imaginally speaking. Okay. But even this, people have considered that is impossible. No way. Why? For the most straightforward explanation, probably what we can say is probably in the case of a van der Waals force, when they have the distance between two carbon, when they have a single bond and when they have a double bond and when they have a triple bond, the distance between two carbons differ. Okay? When they have the single bond, the distance is the farthest. When they have a double bond, they get much closer. When they have a triple bond, they get really, really extremely dangerously close. So theoretically, when they have this, the fourth bond, the quadruple bond, it can even, their nucleus can touch. Two nucleus can touch. So, hey, there's no way of having this bond. Is probably the most plausible and the easy to understand explanation. And that makes sense, right? So, generally, yeah, it's been regarded as an impossible way of. But I asked this morning, I tried the chat GPT. So I asked this thing, hey, why this carbon quadruple bond is not possible, and ask him. And he said, surprisingly, well, actually, it is possible. In 2012 or something, there was the report, scientific research report that's showing that carbon quadruple bond was actually possible. And also there was another occasion that actually demonstrating this bond existence. Okay, so if you thought, and then if you simply just trust 100% what this ChatGPT told you, then wow, then hey, you guys are all wrong. Actually, carbon quadruple bond is actually possible, you stupid. But here is the catch. ChatGPT lies a lot. Why? Because ChatGPT is not something that he or she has a capability of whether this information is really the fact, based upon the fact or not. Simply, it gathers all those available information on the net. Yeah, I actually looked it up myself. That the references. There was in 2012 or something. There was one research paper published demonstrating, arguing that there is actually carbon quadruple bond possible, but it's not proven. A lot of different scientific paper can be published without really rigorous the like the proof reading or the validation, so to speak. So we cannot really trust 100% on their validity or their trustworthy. It could be simply one minor incidence that scientists actually measured or did some wrong experiment. Who knows? Because it hasn't been repeated by any others. So maybe possible and largely still considered to be impossible, the existence of this quadruple bond. So what's the conclusion? Once again, based upon quantum mechanics, then in the world of quantum mechanics, you cannot really separate each individual, these four bonds as a separate covalent bond. So what even you can say is, it may be possible or maybe not possible, depending on different locations. So what about us? While we are living in the earthly world, still we can consider this carbon quadruple bond is not possible because of this specific reason, but still have it ready for the room for accepting. Maybe someday later somebody will prove in our more nobly approach that they can somehow we can explain to us that it is indeed possible. So let's put it that way. Okay. Thank you. Good question. And yeah, this is actually the essence of the enzyme function. Two things come together in the specific area of a specific protein enzyme, and then voila, some magic. And this particular area, this magic occur, we call as an active site of an enzyme that we are going to get into that as part of this. Okay, so I did it. Okay, so okay, fine. So this carbohydrates, simply just ignore all these little details that we don't really need to worry about. Carbohydrate, one thing is they are mostly hydrophilic. That's one thing. Why? Because carbohydrate by themselves is something that carbon, hydrogen, and oxygen. These three things are abundant. And why? What would make this whole carbohydrate hydrophilic? If that's so, this guy. The number of oxygen present in carbohydrates are relatively abundant. So with that abundant oxygen, then there is no other way but oxygen will create some trouble. Okay. It will have a stronger desire for electrons. So polarity will occur. So overall, carbohydrate will be hydrophilic. In other words, why is that something? Why does that matter to us? Because if carbohydrates are such hydrophilic in nature, once again, all we worry about is usually this. Hey. Once again, carbohydrate. Carb. May I enter freely or not? What do you think? Carbohydrate is hydrophilic. Then any given carbohydrate, be able to enter freely or exit freely through cell membrane? Tell me. Think of the case of cholesterol, LDL. What happened? What happened to LDL? LDL needed specific receptor, LDL receptor. Why LDL? Why? Because LDL was hydrophilic. Any hydrophilic guys cannot enter freely in and out here. So any hydrophilic guy we have to prepare some special things for them. So any carbohydrate, we better have what? What is this? R. Receptor. Glucose receptor, sucrose receptor, galactose receptor. Probably we have to prepare every specific different receptor for each different carbohydrate that we are going to import. And also, if they are going to go out in their own entire untouched form, then they better have some specific way of doing so, because they cannot go freely penetrate themselves. That's the reason why we worry about, okay, so any carbohydrate molecule, do we need any receptor or transporter? So that matter, receptor. And another, more important, because carbohydrates are usually used as our like energy ingredient. So in that way, transporter. Without this specific transporter, there's no way we can have an access to. That's probably the more important thing that we need to worry about. And what's the usage? Usually energy. Glucose. Think of the glucose and think of a starch. What's the relationship between glucose and starch? Anybody knows? Starch is probably my favorite macromolecule, probably, for my personal reason, because I love French fries, my the favorite food, and that French fries is a whole bunch of what? Starch, isn't it? But what's the difference between, what's the relationship between starch and glucose? Exactly that one, that monomer and polymer thing. Glucose is the monomer. And using the glucose, starch was made as a polymer. That relationship exists. So, in the case of a starch and glucose, both of them are something that energy related thing, isn't it? One is immediate energy, like by breaking glucose, we extract the energy to make the ATP. What about the starch? It is a storage form of glucose. Right? When you have an excess surplus of glucose, you don't store each individual glucose in the refrigerator of your cell, but you assemble each glucose into starch as a storage form and put them in some specific place, like here or here. That's not the case of starch, of course. A starch can only be made in the plant. So, but we have an equivalent version of our own starch-like things, which we call the glycogen. That's what I'm talking about. So anyway, so all these are individually each different sugars, glucose and fructose, and also ribose, all these different sugars. And in order to build some of the more complex, like polymer, like in the case of a sucrose, what is a sucrose? Everybody knows a sucrose, right? Sucrose, table sugar, white sugar. Sucrose is only two component thing. Sugar is actually glucose plus fructose. But specifically, what type of a chemical bond can you say? To make sucrose, you used what kind of a specific chemical reaction? That in the previous session, what did we? Dehydration or condensation? Exactly right. Condensation, the water. That's why you see this water is out. So this covalent bond, specific covalent bond, we call the dehydration or the condensation, made sucrose. And in the case of starch and glycogen and cellulose, it's no different. Individual things are all built like one by one, making chain through such dehydration reaction. So in the textbook, they have some different classes like simple sugars. These are all monomers in the case of carbohydrates. And And if you wondered, for example, why in some occasion these sugars like glucose is represented like in this structural form, like sort of basically linear, whereas some other times glucose looks like this ring form. Why is that so? Because they exist in both form. Like it, they can shift between linear and ring form in the real world. Although this ring form is predominant one, but you don't have to know this. But both are present and possible. That's the thing that, just in case you are wondered. So, in case you didn't know, all these lines, solid lines, all of them represent what? Covalent bond. So this only covalent bond in chemistry is being represented by drawing solid line. For ionic bond or hydrogen bond or van der Waals interaction, usually we don't put any solid line, but when they are really necessary, then we put some like a dotted line to indicate the presence of ionic or the hydrogen bond. That's what you can distinguish the differences. And so sucrose is a little more complex form of sugars. We call it double sugars, or in more professional way, we call this disaccharide, whatever. Hey, it's me over here. Okay. And so these are all disaccharides, different disaccharide, including sucrose and some others, which we don't have to know. But all of them are the result of specific dehydration, right? Reaction, condensation, like here. I always never miss mentioning whenever we try to touch this sucrose, which is, of course, double sugar or disaccharide, uh, glucose and fructose. But what about this? The chemical structure below, whose name happens to be what? Can you read this? How would you pronounce this? Say it. Sucralose? Sucralose? Sucralose. Great. Yes. Sucralose. Sucrose. Sucrose versus sucralose. Sounds fish. Or, no, I should. Smells fish. Something like. What is this? So what's the difference? Can you tell the difference between sucralose and sucrose? It's a fake, of course, looks like. Fake of sucrose. And then you can easily point out what has been changed. For example, like here, these never occur in the real natural sucrose. So these sucralose are actually synthetic artificial sugar created by in the laboratory, okay, to mimic sucrose. Why do they do that? Because it can, of course, they realized later on, it has a 500 times the sweeter taste than original sucrose, which is higher efficiency. Like you can use this sucralose 500 times less than what the originally sucrose needed in any case. That's a good thing. And better yet, this sucralose is unmetabolizable. That's the point that I need to mention in this classroom. Unmetabolizable. What do you mean by that? So if you use the sucralose instead of a sugar in putting them in your coffee, for example, then probably you don't have to worry about adding any more additional unnecessary calorie to your system. Like if you put 5 spoons of sucrose in your coffee. It may be sweet, but you sometimes worry about, hey, it's too much sugar. So would I get fat because of this sugar that I take? Maybe some of you actually. But here they advertise, they claim that you don't worry about this thing because it will never, never, ever contribute anything on your metabolic system. Why? Why is that? This is the question. Why this sucralose, which is a fake sucrose, would not be metabolized in our body system? Then we have to understand what do you mean by being metabolized in the case of the sucrose. Carbohydrate, yes. Going on. Keep. But this is another type of carbohydrate, although unusual chlorines are added on, but still basically they are also carbohydrate. Carbon, hydrogen, oxygen. So basically their frame is also carbohydrate. But still, like, what do you mean by the sucrose is metabolized? This is a question first that if you answer this, then probably you can answer this too. Because once the sucrose are inside in our body, what happens? Do we store them? No. We usually, immediately we break them down, right? Break them down into what? Individual glucose and fructose. So we break this bond, okay, by hydrolysis, right? Who does that? Enzyme, exactly. There is a specific enzyme responsible for breaking this sucrose down into 2 individual components so that each sugar can be metabolized, means can be broken because they are nothing but energy source, just like glucose and some glucose wannabes. So we are going to extract the energy by breaking them. That's what we mean by metabolizing them. But why can't we? It's the same sugar. We love to break this thing down too. Why can't? Because as she mentioned, this has to be done by only enzyme. That means we don't have enzyme for this. Even though they look so much alike, can you distinguish these two? No. Usually you can, but enzyme can. So enzyme, if we feed the sucralose to this enzyme, original enzyme that metabolizes sucrose, then enzyme will, no, this is not sucrose. I know that. You think I'm stupid? No, I'm not. Right. So that happens. So this will simply pass through without doing anything, simply pass through our system. That's good because it gives what we want, sweet taste, and then leave. That's what actually this company markets. This is good. So, look, probably you can see probably even in like Starbucks, like places like Starbucks, you can easily have some memory of finding them. So this pink thing is the more famous such artificial the sugar like saccharin. Okay. And another thing, the more popular in these days, aspartame. This is something that the resembling structure of a particular amino acid, and this sucrose is like this. This, of course, with a different brand name, they don't have a like sucralose name, but this is a sucrose on everywhere on the such places like cafeteria or anything. Of course, some environmental activist groups warn that sucralose is not something that the harmless, innocent bypass. So it will have some serious problem in your liver. But I don't know. Usually, such claims are like highly blown out of proportion, highly exaggerated ones. So we cannot really put 100%. So probably somewhere, the truth is somewhere in between. Like the developer, the company who create this will advertise, hey, this is perfectly 100% safe. And environmental group is, no, that is something like a second creation of the horrible, the devil, probably in between. Something not 100% safe. There might have been some undiscovered, some side effect. It is always possible. We didn't discover yet. But at the same time, it is not something that we should never try or anything because it's too dangerous. No, there is no such a thing like that. So anyway, our point is why sucralose is not metabolized. Key point is this. Because the same paradigm will also appear again in this chapter 2, again and again. So the enzyme substrate, this is always a specialized interaction relationship, enzyme and specific substrate. So enzyme substrate what? What's the word, the proper word over here to describe this relationship? Enzyme substrate specificity is probably the keyword. They can describe this, explain this. Why? Because for every specific material in our body, there is a specific enzyme which will take care of. That's what we call enzyme substrate specificity. So even with this such a subtle, very tiny modification, enzyme cannot recognize. That much enzyme is sensitive. Or maybe we can say we did not have enough time to evolve yet to take care of such a new thing yet. Maybe given longer time, some of us may evolve some specific enzyme who can metabolize this sucralose, so that individual can have benefit. Hey, I can live on sucralose. You guys can't. Okay, so but until then, everybody has this limitation of this enzyme and substrate specificity limitation is probably the bottom, the take-home message of this instance. And the largest group of such carbohydrate polymer, the examples are like this: starch, glycogen, cellulose, chitin, etc. And starch and glycogen. How many of you do not know what the glycogen is? So everybody knows what glycogen is. Good. These are starch and glycogens are like cousins, very similar. Starch is the one here. Starch is the one made only in plant, by plant. Like the usage, purpose of a starch is storage, energy storage. Like they produce glucose through as a result of photosynthesis. And what are they going to do with this produced glucose? Store them in the form of starch. That's what they do. And glycogen, on the other hand, even very similar in structure, only made in animals. So animal and also fungi. And animal is in particular, they're acting exactly the same role as a starch in plant. So this is a storage form of glucose. So even in animal, there are lots of occasions that you have a surplus or leftover of glucose. Then you just put those leftover glucose together and then build this glycogen as a storage form of your energy. And in your body, In what particular two places can you easily find a whole bunch of these glycogen? Can you name? In your body, two specific area of your body that you can find the abundant, huge glycogen. That means those two areas are the ones that usually mainly synthesize glycogen and store them. One is your muscle, because your muscle, you never know actually when you have such a huge amount of a quick supply of glucose as an energy form. So you better have huge ample storage of such material. So usually you store such energy in the glycogen. And another more bigger area, main area is your liver. Yeah, liver is the place. On the other hand, the cellulose is, one thing is both starch and the glycogen, are the polymer made only of glucose. So maybe starch is the one something that made several hundreds of only glucose. Glucose, all each glucose are connected through that the condensation or dehydration reaction. That's it. Glycogen is two, right? So this is nothing but something very complex, but all you see is glucose. Both the case of starch and glycogen. What about the cellulose? So examples of cellulose are like this. This is a cellulose. And so where can you find the cellulose? Plants have a cell wall. And unlike us, unlike animal cell, plant cell usually have this kind of rectangular shape. Why? Because of this presence of a rigid protective structure called the cell wall, inside of which actual cell membrane is there. So in other words, a plant cell has one additional protective layer. This cell wall is made of cellulose. Whenever you see this OSE, then you know that it must be a sugar, carbohydrate. Think glucose. Or glycogen is something different. Sure. Starch is something different. Sure. But usually the sugars, carbohydrates, simple carbohydrates all end with OSE. Then probably, it's like a sugar. So anyway, cellulose is another such a polymer made of only glucose. What? Then how do you distinguish the starch and cellulose? It's just like apples and orange. We all know that starch is like one of those starch is my favorite, like so that they are always fresh, soft. So we love to digest. But cellulose, you try to digest cellulose, it's not going to, because it's very tough. Cellulose is very tough, rigid. No wonder why this cellulose is being used as a protective layer of plant cell. So what makes this difference? If both starch and cellulose are all made only of glucose, how they make this whole chemical and physical properties are so different, like this. It's not here because you don't really worry about this, but so I have to put my action over here once again to showcase the difference between glucose and cellulose. Oh, what a shame again. So here's the difference. In building starch, glucose. Hey, I'm an individual glucose. So glucose makes starch. What about cellulose? Glucose glucose, 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 glucose. Don't make this one more time, then I will just fall. It's getting dizzy again. So, you get the idea point? The orientation of each individual glucose are arranged differently. Like in the case of cellulose, each individual glucose are arranged alternating orientation. One is facing this direction, and the next glucose is facing the other direction. Whereas in the case of glucose, in the case of starch, and also glycogen too, all each individual glucose are facing the same direction orientation. So that's the difference between chemical bond orientation. Why am I bringing up this? Once again, the enzyme. Why Why in occasion of the French fries, the minute I put one piece of French fry in my mouth, then that French fry will, like, I know that it will be immediately digested. Whereas, even no matter how long I just put this cellulose in my mouth, it will just stay there like undigested. Why? Why is that so? We do have an enzyme for breaking down starch, digesting, metabolism, starch metabolism enzyme. We have it. The name is actually pretty, very pretty. Amy. The name for starch breakdown is amylase. Maybe it's not. Why? I'm not sure the spelling. Anyway, this is amylase. That's why I love this. The name of this starch breaking down enzyme. Of course, I did not have anybody like Amy in my past, in my previous history, any previous intimate history. No, not. But anyway, I love this name of amylase. But we don't have specific enzyme breaking down cellulose. The name is cellulase, of course. Cellulose breaking down enzyme. Cellulase, we don't have, actually. For some reason. This also illustrates that specific enzyme is required for no matter how structurally closely looking component, structurally material, still enzyme will distinguish the difference and only specifically engage or like a catalyze some job specifically. Enzyme substrate specificity is also illustrated. Some microorganisms like bacteria, some bacteria and some other miscellaneous microorganisms have this enzyme. And by inviting such microorganism in our gut, we can display this ability of digesting cellulose. In the past, we were like that. In our appendix was the actual place we used to have this cellulose breaking down a microorganism lived long time ago. It turns out that. But for some reason, we just kick them out. But still some other animals like cows and horses and those animals, grazing animals, still have such microorganisms in their guts. So that's why they can feed on grasses. So they can metabolize or digest cellulose. And chitin is very similar to cellulose. Some minor difference is it has some nitrogen in their polymeric form of this individual unit of sugar. That's all. But more importantly, these chitins are also used the cell wall material for other groups of the organism like fungi. molds and yeasts also have cell walls, but their cell wall material is different from that of a plant in that their cell walls are made of this chitin, not the cellulose. But chemical structure properties are very similar between cellulose and chitin, so that they can serve, both of them can serve as cell wall material. Uh, so those are examples of a polysaccharide, but here, oligosaccharides, what are them? So it's not poly, it's not mono, it's not disaccharide, but oligosaccharide. What kind of difference make their own small group of this recognition? Polysaccharides like starch and glycogen and cellulose, we are talking about several hundreds of monomeric glucoses, okay, maybe even thousand in some cases. Disaccharide like sucrose, we are only talking about two monomers. What about this oligosaccharide? So numbers. When we talk about oligosaccharide, usually the monomeric units on it is made up of something like a 15 to 20 ranges. Okay. So then we call that as an oligosaccharide. And some of you may think these oligosaccharides are some kind of alternative, healthier sugar source, which is being used in reality in these days in our kitchen. But actual, the function of this oligosaccharide in our cell is nothing. So except for oligosaccharides, most of these other carbohydrates, their function, their uses are related with the energy usually, direct energy source or energy storage source. Of course, with the exception of those cell wall material like cellulose and the chitins are structural component. But in the case of this oligosaccharides, this figure illustrates their real function in the cell, which is what? So what are these here? This is what? Come on, say it. What is this? Yeah, thank you. Cell membrane. This is a cell membrane. And then what do you think this is? These two are what? Proteins. They are proteins. So in the cell membrane, like receptors or transporters and all those different proteins are there, like doing their roles of, some are doing the receptor function, some are doing this transporter function, something like that. So many different proteins. But with no exception, all these proteins on the cell membrane have this. What are these? They are carbohydrates. They are oligosaccharides. So when these proteins are made, and then after they are finished, and then these, just like they are having this additional cap or anything like these several units of carbohydrates are added, attached, and we call that as a oligosaccharide. And proteins having such oligosaccharide we call as glycoprotein. Here, glyco means carbohydrate. So this is like a hybrid combination of a protein plus carbohydrate. Glycoproteins, and then protein having the oligosaccharide on top of this. Then what about this? Glycolipid. You can guess. Lipids having some oligosaccharide. These are glycolipids. So in previous slide, you have both glycoprotein as well as glycolipids here. These are glycoproteins, right? And this is glycolipid because these carbohydrates are directly attached to the lipid part. So both are possible and both are happening in our cell is what this figure illustrates. Why is the more important question? Why on earth we have this such ornamental things on the surface of proteins, or in some occasions even the surface of lipid too? One very good example of such glycoprotein on your cell surface, you occasionally actually mention in your everyday life. When you have actually blind date, probably some of you may have this topic as a particular type of glycoprotein on the cell surface. You have never? Not? Oh, surprised. When I was your age, we were actually. We didn't have any too much like topic to share with our prospective partners. So that happens to be one of the very popular topic. What is your glycoprotein topic, the type on your red blood cell? It was like ice-breaking comment between, oh, my glycoprotein on my red blood cell is type A. Oh, mine is too. And so your personality must be blah, blah, blah. Like that. Yes. So this blood type, different blood type, type A, B, and O, and A, B's are all determined by the specific type of carbohydrate on your red blood cell surface. And why is that important? Why is the cell's specific type of carbohydrates are so important in our living? Why? Because, for example, like as a type O blood, me, if I get type A blood transfusion, all those type A blood will what? End up being destroyed. So it's useless. I cannot receive any blood. Why? Because my body defense system, that white blood cell patrols. And then if any red blood cell comes in and they just had this surveillance system, and who are you? How they distinguish which one is mine and which one is yours? By recognizing this. And, oh, okay, so you are one of us. And type A blood, red blood cell came in, and my white blood cell detect, this is not my type of carbohydrate, so you are not one of us. So I will destroy you as an invader. This is what happens. So recognition. How do we distinguish which cells are which and those things? They're all being relied upon by this specific type of carbohydrate, which is very important. So that's why all the proteins on every cell's surface have, all of them, without any exception, have very specific type of special, different type of carbohydrate in the form of oligosaccharide, and which is very important in providing as a kind of, hey, it's me type of marker. Cancer cell, at will, they alter this type of specific marker. And that's why white blood cell can detect, oh, that guy is something like a cancer cell, and they destroy. And cancer cells, so in response to such defense mechanism, they have some other mechanism of altering such surface marker. It's all battle. All ongoing battle is going on even between cancer cells and our body defense system too. So, yeah, there are lots of things that we can talk about, keep talking on and on about this oligosaccharides functionality, but just let's put it, yeah, their overall the utility or function is not for energy or protection, unlike other carbohydrate, but this recognition is okay. Lipid. Oh my gosh. So I get to only finish only the lipid. I cannot even finish lipid here. Shoot. Can you give me 10 more minutes? Or you want to go home? You tell me. I will follow. I follow your order. If you say yes, 10 more minutes, then I can try a little more about lipids in within 10 minutes. Or hey, no, whatever. What the? Go hell with the lipid. Then I will just let you go home and then worry about the lipid next week. Just you decide in 10 seconds. 1, 2, 3, 4, 5. If nobody say yes, then anything, then I will just go ahead and have 10 more minutes. 8, 9, yeah. You saved entire classroom. Okay, so everybody should thank to this lady. Okay, see you next week.